Perfect. Thanks very much for that introduction, Laura. So I suppose the title of my PhD and the work that I'm going to be talking through today is an evaluation of antibiotic-resistant Enterobacterales in the Irish environment. So although I am lecturing in ATU Galway, my PhD was carried out in the University of Galway. So to begin with some introduction and kind of background on antibiotic resistance, and I suppose just to mention throughout the slides, I've included some nice images of different kind of regions where we sampled as part of the PhD. So I think it's nice way to break up some of the content in the presentation. So antibiotic resistance is a global public health challenge, and it's probably something that most people are aware of, whether you're in the kind of scientific field or not. And there is an estimated 4.95 million deaths associated with bacterial antibiotic resistance, which occurred worldwide back in 2019. So this was kind of a systematic analysis that looked at the rates of deaths kind of worldwide and linked those that had kind of an underlying resistance association. So in terms of, I suppose, what kind of pathogen drug resistance combinations cause these deaths, so somewhere in the realm of between 50,000 and 100,000 deaths were contributed and caused by each of the following. So I suppose the reason why I'm flagging these is because you'll see when I go into kind of the results of our research, we did find some of these isolates and bacterial species in the environment as well, and particularly isolates that displayed these types of resistances. So the first was E. coli that were fluoroquinolone resistant and third generation cephalosporin resistant, and the other type was Klebsiella pneumonia that was carbapenem resistant and also third generation cephalosporin resistant. So again, these are just different, I suppose, classes of antibiotics that would be very important for the treatment of infections caused by E. coli and Klebsiella. So something that I suppose I'll mention and an interesting part, I suppose, of our findings in this work was the detection of something called CPE, which is a carbapenemase producing Enterobacterales. And really, I suppose these are important to flag because they were declared a public health emergency in Ireland back in 2017. So they are a very, I suppose, significant problem in terms of the treatment of infections because the carbapenem class of antibiotics would be considered one of the last resort classes. So any resistance mechanisms against that particular class makes our treatment options very, very limited. The most recent data in relation to the kind of clinical aspect of things, so the Galway Reference Laboratory, showed that in 2020, we had 694 new cases of individuals colonized or infected with CPE. So in the case of colonization, the individual may not display any symptoms and they may not realize that they have this type of bug, but they can spread it in their fecal material. And of course, people that were infected with CPE pose, as we said, a very serious risk in terms of, limitation of treatment options. So in terms of the types of CPE then that were isolated, this table again was taken from the most recent HSE reference laboratory report. So along the kind of left-hand side, we can see the different bacterial species. So again, lots of E. coli, Klebsiella, some Enterobacter and Citrobacter. And along the top of the table, then we can see the different types of carbapenemase genes that were detected. So again, for example, I suppose just to highlight some of the more common ones that we see, such as our OXA48, the KPC, we also see NDM and OXA181 very commonly in circulation in the clinical environment. So recent reports have recognized that antibiotic resistance is a One Health challenge. So although we have, I suppose, lots of data relating to the clinical environment and from a veterinary perspective, there's not much information in relation to kind of antibiotic resistance in the environment. So there is an increasing need to better understand the complex role the environment plays in the spread of antimicrobial resistance. And I suppose for anyone that hasn't heard of the concept One Health before, it really is a kind of a way of recognizing that the health of humans, animals, and the environment that they share are all interlinked with one another. So in figure one on the right, I've included some examples of kind of the interactions that we see between humans, animals, and the environment. So again, you can have a closer look at that image, but again, there's, I suppose, loads of different ways. If I to kind of zone in on one, for example, if we think of healthcare facilities where individuals would be exposed to, lots of antibiotics in their wastewater, often we would see high levels of antibiotic resistant bacteria as a result. In Ireland, we have things called stormwater overflows. So a lot of kind of stormwater would go into our drains that collect and kind of transport wastewater. So if we do have quite stormy events, this we would see discharges of this wastewater that's untreated. In Ireland, we also have raw sewage discharges, unfortunately, and these are, I suppose, being taken care of by Irish Water. And the eventual hope is that we won't have any, come a couple of years' time. But again, I suppose there's lots of different ways that these kind of resistant bugs can get into the environment. And then, of course, we, interact with the environment through, recreational activities such as swimming and surfing. We consume different, fish products and all of that. So again, I suppose everything really is interlinked in this One Health triad. So moving on to kind of the aims and objectives of this work. So our aim was really to look at both sewage sources and water bodies across Ireland and screen them for the presence of antibiotic-resistant Enterobacterales. So just to mention, Enterobacterales are an order of bacteria and include species such as E. coli and Klebsiella that would be very common causes of clinical infections. So we collected samples and looked for the presence of extended spectrum beta-lactamase producers and also carbapenemase producers. So again, these are just, I suppose, a types of beta-lactamases that can break down beta-lactam antibiotics. So really, they do severely limit treatment options for people that are infected with either ESBL or carbapenemase producers. So we collected samples, I suppose, across Dublin, Cork, and Galway, highlighted in the map in the right. And we were lucky enough that we were able to look at some of the isolates at a genetic level. So we did carry out whole genome sequencing, and we were trying to kind of compare particularly the isolates from sewage and waters to see if we can establish if there's kind of a genetic link between the two of them. So in terms of, I suppose, the publications from this work, we did publish an initial point prevalence survey. So this was our first round of sample collection across the 3 counties. And you'll see the QR codes for both papers on the slide if you are interested to read up further on it. But in, I suppose, the interest of time for this presentation, I'm going to focus on the results of the longitudinal survey. So this was an additional two sampling rounds across the 3 counties in Ireland. So again, I suppose what we found across both rounds were relatively similar as well. So I'll highlight the key findings there. So in terms of the longitudinal survey then, this included, as I said, 2 rounds of sample collection. So we were looking at 118 water samples and 36 sewage samples. So the table 5, I suppose I'll just mention that first on the right-hand side. It shows a breakdown of the different sample types collected. So primarily seawaters were the most common type of sample. And, in order to, I suppose, select the sites where we would collect samples, we did create maps of different types of sources that can, discharge potential antibiotic-resistant bacteria. So we looked at, discharges from wastewater treatment plants, raw sewage discharges, and all of the aspects, I suppose, that could contribute to the spread of antibiotic resistance. In terms of other sample types, then we also looked at rivers, lakes, estuaries, drinking water treatment plants, but we solely looked at the influent of these. And then we also looked at a number of sewage sources as well, such as hospitals, nursing homes, airports, and wastewater treatment plants. We isolated a number of different bacteria from these samples. So the most common ones that we did find, interestingly, were E. coli and Klebsiella. And then to a lesser extent, you'll see in table 4, we've also found some Enterobacter, Citrobacter, and so on and so forth. So some of, I suppose, the key findings, again, this is a very quick snapshot of this work, so do refer to the paper if you want more detail. But we did identify ESBL producers in 79% of the waters and 50% of the sewage samples. So they were very, very widespread across particularly the water environment. A total of 18 waters and 5 sewage samples harbored one or more CPE. So again, this is a type of bug that is conferring resistance to some of the last resort antibiotics that we have. Interestingly, we also saw that 4 isolates harbored MCR9. So if anyone's not familiar with what that is, it's a mobile colistin resistance gene. And a colistin would actually be one of the antibiotics that's used to treat CPE. So it's even one of the more last resort of the last resort antibiotics. So we also looked at the plasmids, and this is a type of mobile genetic element that can kind of jump around from different bacteria and spread antibiotic resistance genes. And we found that the majority of isolates did harbor one or more plasmids. So again, indicating that they have good capability in the natural environment to spread antibiotic resistance further. So figure 6 then on the right-hand side is a core diagram where I've kind of shown, I suppose, the different sample types along the bottom half of the circle. So you see the likes of the hospital, for example, highlighted in yellow, and the corresponding carbapenemase genes that we identified across the different sample types are along the top of the diagram. So again, for example, if we look at the hospital sewage sample, we can see that we identified KPC2 and OXA48 in this particular sample type. So again, it was just, I suppose, a nice way of kind of summarizing the information and presenting it in a visual format. So some other, I suppose, interesting findings, particularly just looking at our CPE isolates. And again, we've broken these down to the different carbapenemase enzymes that they produce. So OXA48 producers, NDM, and KPC. So this figure includes an antibiogram of the CPE isolates detected. And what this is we set up a panel of different antibiotics and tested whether the isolates were susceptible, intermediate, or resistant using our UCAST breakpoints. So in this case, you can see there's lots of red, I suppose, that stands out in this particular image, and particularly for our NDM and KPC producers, you can see that there's a very limited arsenal of antibiotics that were actually susceptible and could be offered as a possible prescription and treatment option for these isolates if they did happen to cause infection. So it is quite worrying that we are seeing such high levels of resistance in the natural environment. So again, this is outside of a clinical environment where you might be assessing clinical isolates. It might be almost more expected, but in the natural environment, it was quite a shocking finding. The final aspect of the results then to mention very briefly is in terms of comparing our isolates at a genetic level, we decided to compare them by examining the core genome multilocus sequence types. So in doing this, we looked at a number of different loci in the E. coli and the Klebsiella isolates within the core genomes of the isolates, and we checked how similar they are to one another. So you'll see there's a minimum spanning tree illustrated and listed as figure 8 on the right-hand side. And each individual circle relates to an individual isolate. So in the case of isolates that are identical, you see the circle is divided in half, or it can be divided in thirds, depending on how many isolates are identical. And the length of the individual, I suppose, lines between them and the numbers that are indicated there indicate how many loci vary between them. So again, it just is a good indicator of kind of clusters and seeing kind of if we do have isolates that are quite similar on a genetic level. So in terms of the clusters for E. coli, we saw some from seawater and a wastewater treatment plant influent in Cork, and a nursing home and an estuary sample in Galway. So we did see some isolates from wastewaters and from waters kind of in the nearby area that appeared to be genetically identical. So again, leading us to believe that they potentially may have originated from the sewage source, and that would have been transported into the water. In terms of our Klebsiella, there was 3 kind of clusters that arose of interest when we did this type of comparison. The first cluster was samples from river and estuary in Cork, and this actually spanned a distance of 9.8 kilometers. So again, very interesting to see that, isolates that were collected from, a large enough distance apart were actually appearing to be genetically identical. So indicating, long-term persistence of certain bacterial isolates in the environment. We also, in cluster 2, saw identical isolates from a river and estuary sample in Cork, And in case of our cluster 3, we actually found identical isolates in a river in Cork, but also in a wastewater treatment plant influent and estuarine water in Galway. So a little bit hard to explain that when there's such a large geographical spread between the sampling sites in that case. To finish up then with the kind of, I suppose, discussion on kind of what we found and recommendations, I suppose, that we would give based on our findings of this work. So in summary, we did see a repeated detection of CPE in aquatic environments, and this was particularly across urban areas. So we did collect samples from hotspots and kind of colder spots that don't have as many discharges into the environment. So this does indicate that there is a potential anthropogenic origin or kind of, due to maybe human discharges. And also what was very interesting is that there were very similar resistance types circulating in the clinical and our natural aquatic environments. There is knowledge gaps relating to colonization of antibiotic resistant organisms caused by exposure via the aquatic environment. So what this means is, we don't know whether if individuals are interacting with the environment through kind of swimming and surfing and those types of activities, whether they're at a higher risk of kind of swallowing the resistant organisms and then remaining in their gut. So this is, I suppose, an important knowledge gap that my research group is currently working on. In terms of CPE detection, then in the sewage aspect, we did detect it in hospital sewage, in wastewater treatment plant influence, and also in airport sewage. So it begs the question as to whether we should be using, kind of a surveillance system, particularly in the likes of our wastewater treatment plants, which we've seen has been very useful in the case of COVID in kind of detecting the levels in the community. So should we be applying a similar approach when it comes to resistance? Similarly, emerging threats, if we look at the likes of airport sewage, is this a way that isolates that are resistant in other regions of the world are being introduced into our environment? So again, this could help us identify if there are emerging threats. And in terms of hospital sewage, is on-site treatment, would that be a preferable option because it would prevent the bacteria from mixing with other environmental bacteria and kind of preventing that spread and dissemination of resistance genes using things like plasmids, as we mentioned earlier. So in terms of our core genome MLST comparisons, again, we mentioned that we did see similar isolates across water samples and also across water and sewage samples. But the gene transfer mechanisms amongst aquatic isolates is an important knowledge gap. So we don't know, I suppose, the ins and outs of how far resistance genes and things like that can spread across aquatic isolates. Ireland's wastewater management system does require intervention. So between the years of sample collection for this project, 2018 to 2020, the number of raw sewage discharges only reduced from 36 to 34. And the most recent report that outlines the number of discharges in 2021 shows that this is only reduced to 32, I believe. So there's only been a further reduction in two sites. 12 wastewater treatment plants weren't compliant with the EU's treatment standards in 2020, and we also had a number of discharge events from the sewage network during the bathing water season alone. So again, in the case of kind of storm waters, we would see that, the capacity being reached in terms of the sewage network. So we would see that release of untreated discharges as a consequence. And what's also interesting is that, across Europe, there's very limited monitoring of the aquatic environment for antibiotic resistance. So we do need to, I suppose, decide on a more harmonized approach. Now, this is, I suppose, it could be a whole talk on itself in terms of, defining an ideal marker. But one really nice example of a potential route that you could go is the Tricycle project, where they looked at just looking for the presence of ESBL producing E. coli across human, animals, and the environment to be able to gather data that's comparable across the three. So again, to see what kind of isolates are in circulation across all aspects of the One Health triad. So there's future work ongoing as part of the ARREST project. So we are looking at comparing the environmental CPE isolates to those from the clinical environment. So again, to be able to link whether the isolates in the aquatic environment may have originated from human sources. We're also doing further investigations of mobile colistin resistance harboring isolates. And the preprint for this paper was released earlier this year, I believe. So we are hoping to submit it to a journal shortly to get a proper publication. Also, there's risk ranking analysis relating to environmental exposure ongoing. We're also participating in a shotgun metagenomics study where we're looking at the complete DNA content of the environmental samples rather than using a culture-based approach to identify bacteria. And then more generally, we do require a standardized methodology for routine surveillance of environmental antibiotic resistance. So again, something like the Tricycle project, as I mentioned, where we see kind of a marker that can help us, compare across all three. And finally, the nexus or the connection between recreational environmental exposure and colonization with resistant bacteria does need to be investigated further. So I'll just finish up by saying a big thanks to everyone that was involved in the arrest project, and I have them listed on this slide here. And also a big thanks to CIWEM for inviting me to present at this webinar. So I won't take up any more time, and I look forward to answering any questions you guys may have at the end. Thanks a million.
Thanks a million for that, Brigid. That's really interesting. I have some questions myself, but I'll keep them to the end. So now Conall should be there. So Conall has also finished his PhD. Conall now works in industry in NVP Energy, located in Blackrock. Well, Conall is located in Blackrock, but he did his PhD over in the West Coast, I believe.
Yeah, in the University of Galway. So
University of Galway,
I'm just going to throw the share here. Yes, thanks very much, Laura.
No worries, thanks, Conall.
Presumably that's popping up okay for everyone. So my name is Conall Holohan. Similar to Brigid, I also did my PhD in the University of Galway. I think we were neighbours for a while. I was in the Department of Microbiology. So I suppose my PhD was on looking at fat and its degradation through anaerobic digestion. So I suppose the simple point to make is that fat for bacteria is also difficult to break down, similar to ourselves in humans. So what we looked at is the degradation of fat through anaerobic digestion into energy. And we looked at microbial solutions that are currently on the market, and then how to bring them into the next, I suppose, the next phase, the next generation. Overall, we worked together with a large group of people and kind of came up with a network to not only define my own project, but also to define how lipids and fats are utilized across the board. So we see, I suppose, lipids everywhere from your milk factories through to your fat, oil and grease traps in restaurants or in big wastewater treatment facilities. And how is that used? So currently at the moment, a lot of fat is generally just mixed in with the aerobic sludge that's produced in wastewater treatment plants and forgotten about, or even it's just put into incineration. And I suppose to state the obvious, fat obviously is an extremely valuable energy source. It's nature's energy source, nature's energy store, and from that we want to make sure that we use the most of it. So our project and the overall kind of scope that we wanted to bring forward and through this presentation and a publication that we can share afterwards is how to utilize fat in all its forms and what's the most efficient. So we looked at it from a very broad sense all the way down to the microbial genetics, similar to what Brigid was doing in looking at sequencing and how the genes of the bacteria that were being utilized. So what's the problem? Why do we have a problem with fat, and what is the issue? So I suppose simply put, fat is degraded from lipids into glycerol and what we call long chain fatty acids. And those long chain fatty acids cause issue to the bacteria. Ultimately, the bacteria struggle to break them down and it causes inhibition. So if you have a dairy factory and you decide to put all your cream into the wastewater treatment plant, you'll soon find that the fat will bind and will cause issues for the bacteria. Similarly, if it's an aerobic or an anaerobic system, if you just put the fat straight into a generic, let's call it, wastewater treatment facility, you will have issues. So, and what those issues are is ultimately the long chain fatty acids binding onto the bacteria, and the bacteria archaea not being able to degrade them quick enough, and you getting that negative feedback cycle. So what we've done is we've gone and we've looked at how can we degrade these LCFAs a bit more efficiently? What do the bacteria need? What kind of environment do they need? And do we need to grow certain types of bacteria? And kind of across that board. So I suppose the issues there are very simple, is you get a certain toxicity, albeit it might be temporary, and we now kind of deem it temporary inhibition. So there's been work on this since the 1980s all the way up to in the last 5 years, whereby we now kind of view the inhibition from long chain fatty acids and their absorption into the bacteria as temporary inhibition. But ultimately that temporary inhibition can lead to the failure of an engineering wastewater treatment plant. Biomass or the bacteria themselves obviously can float if they are surrounded by fat, similar to what you would see fat balls in oftentimes being if you have a serious fat incident. And also then we look at granules, which are a certain form of, you might know it from the Nereda system in aerobic systems, or anaerobic granules in anaerobic systems. And I'll talk through that in a little bit more detail. So like I said, from the 1980s up to the, I suppose, the recent years, there's been significant breakthrough points. And we've outlined that in this perspective paper we put together. But I suppose from that, we try to bring forward what are the knowledge gaps for the scientists to keep working on, but also what are the jumps forward and the leaps forward that have been put together by the scientists. So now as engineers, and I'm sure a lot of you are in the wastewater treatment sector, can actually utilize that and bring the gains forward on a day-to-day basis. So like I said, fat is an issue, but if you don't use it, you ultimately waste a lot of energy and a lot of potential resource in a society now where we're really aiming to achieve resource recovery. I think that's something we should all take into consideration. So on this slide with a little bit of funkiness, you can see there just very simply what happens with fat containing wastewater, both on an aerobic system or an anaerobic system. Ultimately, it's removed. And you'll often find it being removed by a dissolved air flotation system and just disposed of in land spreading. Now we're seeing, I suppose, coming from the Netherlands, that land spreading is beginning to be phased out, and that will ultimately lead to incineration. But again, you're losing that energy. You're losing the energy from the fat, and because of an inhibition that we, I suppose, would like to make the point here that can be overcome. So let me just click forward. So I suppose to give you an idea as to the value of fat, if you compare it to the other parts of wastewater, be it carbohydrates or protein, for each gram or kg of the substance of carbohydrates, you'd only produce a minor amount of energy, or protein similarly just less than half of that for the same amount of fat. And that comes back to the natural idea that fat is an energy store ultimately for biology across the board. And there in the graph, you'll see the difference between a dairy factory, an Irish dairy factory that we have an anaerobic digester on, treating the wastewater, that if we accepted the fat, you would get significantly more methane. You're talking up to 3 times as much methane to the factory as you would when you remove the fat, which is in red. So it's very simply the calorific value of wastewater in sites that have a lot of fat is significant. So I suppose that lays out what should we be doing, which is very simply, we should be using the fat as directly as possible. So ideally not removing it from the wastewater and using it directly in anaerobic digestion while you're cleaning your wastewater. So this is our kind of proposal, this fat through anaerobic digestion to energy biotechnology. So that was essentially my PhD, but again, I'm trying to put it in the grand scheme of the field to you here today, but ultimately to give you a tangible idea of what are we looking at here. So obviously the Irish economy is based a lot on dairies, and these figures are from 2020. So I'm sure they've even increased since then, but ultimately about 375 million litres of waste dairy fat are produced. So that would be fat that's removed from wastewater around the country. And that has the potential, if utilized directly, to produce a significant amount of biogas equivalent to about 400,000 cars over the course of 10 years. So it's a significant lump of potential bioresource. So I suppose, why aren't we using it to date? And that's very simply because our anaerobic systems, and I'm using anaerobic because you get your energy directly back through biogas, aren't capable of doing that. So there's several different anaerobic filters or anaerobic digestion systems for wastewater, and we tested that during my PhD, and I kind of proved or reproved that they would fail with lipid-rich or fat-rich wastewaters. So the first generation of systems, we don't have a huge amount of them in Ireland, but you will see them across the UK and Europe, which are granular biology-based systems, UASBs and EGSBs. And the key limitation there is your fat wraps around your bugs and ultimately inhibits them, and they get washed out, and you don't grow your bacteria quick enough. So from that point of view, you always remove your fat if you're going to be utilizing one of those systems. And our hypothesis very simply was, how can we overcome that? Now, since then, I suppose the anaerobic field has grown and grown and grown over the 80s through to, this is up to 2017, but continues to grow very simply due to the generation of biogas and its values increasing. So you can see here that the growth of a third generation is coming forward, whereby the development of higher rate systems and systems that are able to deal with more complex wastewaters are coming to the fore with some of the companies that you might recognize. So this is a commercial example of two systems that have the ability now to treat lipids, and we'll kind of talk through them briefly. Also, one is the PAX AF4, which is a flotation-based system. So this bypasses using engineering, and similar to that, the Spartan system also bypasses engineering, upflow and washout of bugs that have fat wrapped in them. So what they use is flotation as a benefit, and they capture your bacteria that are remaining. And then they ensure that you can reuse those bacteria while still degrading the fat, and they allow more time, but you're into a larger system, and you still have your systems that might be on sites already that still can't utilize their fat. So while there's a benefit to them, and if you're going to a greenfield site, that's positive, doesn't fully, I suppose, allow you to use what you have there already. So like I said, there's also lacking, I suppose, a clear understanding of the fundamentals governing the LCFA degradation. So why are we getting inhibited? Why can't we kind of power through that inhibition? And then I suppose we have also looked at the morphology and the presence of the overall bacterial communities and their acclimatization to lipids. And I'll kind of talk through that now. So focusing on my own research, what we have looked at through, I suppose, actual lab research over the course of 3 and 4 years is developing a bioreactor that can indeed treat lipid-rich wastewater. And this is just a quick pic from the lab, but you can see there, you can see tall, skinny reactors with your bacteria at the bottom. That would have the ability to treat lipids in the wastewater. And that's based on two approaches. One is an understanding of the bacteria that have gone in there and utilizing an acclimatized bacteria. And the second of all is ensuring the retention of the bacteria without washout, and while allowing for degradation of the fat. So I suppose just to give you a quick view of the results, apologies, a little bit of shifting on the PowerPoints, but ultimately you can see here in the top left the removal of the COD, which would be the chemical oxygen demand, just as a marker of how much pollution is in the wastewater, and indeed the suspended solids, whereby you get a clear removal rate. And I suppose from a point of view of utilizing TSS is that you don't lose your bugs through that, and that will be viewed as TSS. The organic loading rate, then obviously we would have ran it over the course of about 110 days, increasing gradually until, and this was utilizing real dairy wastewater with fat still in it, that it does fluctuate over the course of that time, and hence the methane quality and methane percentage will fluctuate slightly also. So we ran this over about 100 days, and this was again a design whereby we had a filter system within the reactor to retain our bacteria, and that allowed for, I suppose, both a retention of the bacteria, but also a secondary cleaning phase as it went up through it. And this is just a quick snapshot of proof of concept, but I suppose, and as is published, and we can send out the publications afterwards, this is a possibility due to, I suppose, longer, more focused microbiology studies. So I suppose I want to give you an overall view of what's there available on the market, not just the research that has been done by ourselves, and what you can do if you have a lipid-rich wastewater. And I suppose you now utilize DAF separation most of the time, and that those DAF solids can go through to low-rate agri-AD, if that's possible, or indeed, I suppose, from that point of view, it can go to mix in with sewage sludge, and that is a positive use of it. However, you have a significant amount of transport involved in that and logistics. The ideal would be to utilize something directly on site that is on the market potentially. However, I suppose the systems that are currently on the market have the issue of needing longer retention times or degradation times, and therefore have quite large tank size. However, they do bring the significant benefit. We have one down in Dairygold in Ireland. Those of you might know it, significant land space, but they are a robust point of view. And I suppose our point of view is to bring forward the idea of directly treating and maybe having an additional piece of kit or technology brought in to the previous generation, so that you can, and our suggestion is a filter system where you can utilize that, so you can directly have utilize fat on site without having to build a very, very large system. But ultimately, I suppose the point we're making is lipids are valuable and they should be utilized where possible. Like I said, you know, the reality is fat is removed from wastewater across the country and across Europe, and we all have an AD system, well, not so much in Ireland, but hopefully in due course in the community, and utilizing what we call co-digestion, which is digesting sewage sludge or manure with another product that gives an increase in methane production, is something valuable. And lipids are ideally placed to be that co-digestion substrate because they are so energy rich. However, it does come with the in-between of trying not to step over the line of temporary inhibition. So I suppose in the paper we published, there's a little bit more detail here, but there is what we have tried to outline is kind of that threshold of the methane yield compared with the loading rate or the amount of fat you would put in. And there is that, it's in the diagram there, depending on what you're mixing and how much you're mixing, but there is the idea that you can don't push it past kind of 2 grams per VS per liter per day. But yeah, happy to discuss that offline with anyone who has the idea of trying to push their methane yield. So just to kind of wrap up and give you a little bit of perspectives on how we view things. So we're very much just in this overall perspectives and as on the back of my PhD, trying to bring the reality of lipids is a valuable waste, but is often forgotten because it just was an issue for so long, and therefore it was just mixed in and forgotten about. And sometimes you might be lucky and it was mixed in with something that's going to the AD systems, but oftentimes it's forgotten about and just land spread, and in the future will be incinerated. So from a policy point of view, we recognize that we should utilize this, and maybe hopefully we would like to see it being fed into future policies, both at a company level or then obviously at a government level, that substrates, that waste substrates like this, while small in volume, can have a reasonably big impact. And I think we kind of showed that with our example of an actual dairy site giving 3 times as much methane back from their wastewater if they utilize the lipids. Training is something that is kind of often forgotten, and it's just the use of the correct technology, that we're not always going to the aerobic technology or the basic AD technology, that there are other options out there, and that there are, you know, innovations always being pushed. But actually a lot of the what might be viewed as innovation in Ireland or in Europe is well established somewhere else, and that gives significant payback, and especially in an environment where gas is extremely valuable. And then naturally we have outlined scientific excellence and ultimately the knowledge gaps that still remain, which primarily are based around the understanding of how the bacteria gets inhibited, and is there a way of looking at that from a microbiological point of view. So if any of you are looking to put in grant applications in the near future for a bit of microbiology work, there is some nice ideas within it. Yeah, so I suppose a little bit of closing gaps. I'll flick past this in the interest of time. Thanks to all my collaborators on this paper and across the board, across NUIG, and I did significant amounts of this research in the Netherlands in Wageningen University. Science is very much a team effort, so it's very much on the forefront of this is the lots of collaborations we have working with us over the few years. Thank you very much.
Great, thanks so much for that, Conall. Very interesting. Some of it a little bit over my head, but very interesting. So we have a couple of questions in the chat box. Conall from Owen O'Flaherty, are there any notable impacts of lipid-rich substrates on biogas quality compared to conventional biogas?
Simple answer is no. I guess we find that the biogas quality in wastewater, anaerobic wastewater systems is about 20% higher. So you'd be looking at an 80% methane rate compared to 60% when you're treating sludge or let's call it co-digestion of food. That is not impacted by putting lipids in. You don't get a reduced methane value. It's more methane with, I suppose, better quality substrate and better quality bacteria in your systems for wastewater treatment.
Great, thanks, Conall. And another question, so from Eduardo Piano, sorry, Eduardo, what is your view on the use of pre-treatment technologies for this type of waste, whether a form of enzymic hydrolysis or thermal hydrolysis?
That's a very interesting question. A little bit controversially, I'd be inclined to always try and go a direct route. However, if you do have lipids, let's say removed from wastewater already, you have quite a high valuable and concentrated substrate. We looked at thermal based and microwave based systems previously with collaborators of ours, and there's more work on this being done by a new or reasonably new PhD in Vincent O'Flaherty's lab in microbiology, UChenley. But not to preempt his results, but we find that you just have to be careful with the amount of energy you're putting in to the amount of energy you're going to gain. There will be a certain amount of degradation anyway. It's just the amount of lipids you could add into a small space that might be increased. And again, with enzymatic, is how readily available are those enzymes will be the qualms, but not to be negative or anything. Everything is possible, and if it adds a positive jump in your methane potential, then absolutely. It just has what I would say be tested on an individual basis, not to be dodging the question.
Great. Another question there for Conall from Rami Kremesti. How does FADE work? Is it patented or open source technology?
Our technology would be based on the back of the technology previously patented by NUI Galway. It's patented and licensed by MVP Energy. But ultimately, it's a two-phase system that utilizes the bacteria like a granular bacterial system as normal with the USB, EGSB system, if you know them. And then it has an anaerobic filter segment that's part of the patent. But there are unfortunately, I suppose, from my own point of view, that most technologies get patented these days. But there are ones, I suppose, you can utilize the ones that are off patent as well. But yeah, happy to have a discussion if you just reach out and point you in the right direction.
Right. Another one there from Rami. Also, the COD graph showed 90 days most efficient degradation time. Is that not too long?
The degradation, I suppose, is viewed in two forms. So the soluble degradation, or the degradation of soluble COD, should be happening essentially immediately and quite quickly within a matter of hours. However, fat is not solubilized most of the time, so you have to retain it as a solid, and then that degradation time can take days. But if you have enough bacteria, so that bacteria, there's always enough there to degrade the soluble, while also converting the solids into soluble. You have a kind of a two-phase system whereby the solids are being slowly degraded and retained, and you're not washing them out, and then your soluble is just going in the background. So it's a two-phase approach.
Great, Conall. Brigid, I have a question for you. Actually, I have three questions. The first is, like, your research is clearly very interesting and very relevant. I suppose, how do you see it maybe integrating into industry, or where might it go from here? It's clearly very powerful information, but how is it going to make its way forward?
Yeah, I suppose it's a little bit different in the sense that we didn't develop any new technology, but I suppose the results kind of highlighted the damage that we're doing to our environment. So I think the removal of, like I mentioned, the raw sewage discharges, even looking at capacity of wastewater treatment plants and trying to avoid those kind of stormwater overflows would be really, really important as well. So yeah, I don't know. I suppose I'm not in the industry kind of field, but I don't know if that answers your question. I suppose it kind of highlights that, we probably do need to monitor our environment and figure out ways to kind of protect it more than what we're doing currently.
Thanks, Mel. Also another question there. So from Tristan Nolan, this is for you, Brigid, as well. Do you think the environmental analysis of AM or microbes in wastewater treatment plants and lakes, etc., can offer valuable information such as regional resistance, similar to SARS-CoV-2 in wastewater treatment plants?
Yeah, this is an interesting question. I think I touched on it very briefly. I think it would offer valuable insight. I think there was a study that looked at kind of resistance in the hospital environment and kind of a local wastewater treatment plant, and they did find kind of higher levels of resistance in the treatment plant, things that they weren't picking up in the hospital. So it kind of encapsulates, I suppose, everyone in the community, as well as because we have so much data, I suppose, in relation to kind of clinical isolates. When people go into the hospitals, they'd be screened for colonization and infection. In terms of a veterinary perspective, if animals are sick, we collect samples as well. So I think some kind of monitoring and wastewater treatment plants would be an absolutely brilliant one, I suppose, to start with, and to kind of evaluate, what's going into our environment. Now, I know the wastewater is being treated, but often the technologies used aren't kind of focused on breaking down genes in particular that harbor these resistances. So hopefully that answers the question.
Thanks, Brigid. Another one here for you, Brigid, from Rami Permesti. And why don't hospitals disinfect their wastewater at source? Is there no legislation, no requirement, no knowledge, or?
Yeah, it's a good question. I suppose it's not used currently in Ireland. And there's no kind of on-site treatment as far as I'm aware for kind of the likes of hospitals and nursing homes. So it's kind of, I suppose, presumed that it'll be dealt with at the wastewater treatment plant once it flows there, and it'll be treated at that point. But I suppose, not as many people consider that damage could be done at that point in terms of the spread of antibiotic resistance genes and introducing it to environmental organisms as well. So I don't know why it's not done. I suppose probably from a cost point of view, it might not be considered as important to implement in the individual sites, since people do argue that the wastewater will be treated at the wastewater treatment plants once it reaches it. But as I said, there's kind of a number of issues with that in relation to the stormwater overflows and it being released prematurely. So hopefully that answers the question.
Thanks, Brigid. So I think one more and then we'll all head off for our pancakes. A question for Brigid from Edardo Piano. Apologies again, Edardo. The AMRs that we're seeing, are they from veterinary or human sources or both, or is it possible to tell?
Yeah, that's a good question. It is quite difficult to link them. Often we look at things like sequence types, and we might see, for example, E. coli sequence type 131 would be very prevalent in the kind of clinical environment. But you would kind of have to take the approach similar to what we've done, is kind of take sewage samples, take water samples, and try and compare them at a genetic level to get a more accurate kind of, I suppose, idea of where the isolates in the water may have originated from. So hopefully that answers the question.
It's great. And I think that's a wrap, Brigid, because I know you have to be elsewhere at 7 as well.
Perfect.
Great. Thanks, everyone. Thank you so much, Brigid. Thank you so much for really interesting, presentations.
Thanks very much.
Thanks very much, Barbara, for helping organise and chaperoning our session as well.
Well, thank you to the branch for arranging this, and two really, really interesting talks on different topics, but interesting debate of our wastewater. There's global initiatives as well with all these things, so it's really excellent to hear about them and see the cutting research that's been done. In terms of the recordings, are we able to publish?
Yeah, that's no worries.
Excellent, right. We'll try and get those up in the next couple of weeks, the recording published in the next couple of weeks. I put in the link to our YouTube channel if you want to see this recording of this webinar or any others that we've done recently. So I really, really thank you, and look forward to seeing you again.
Thanks very much.
Have a lovely evening. Enjoy the pancakes.
Take care now. Bye.
Bye now.
Bye.
Thank you.
Bye.